All right, moving down, we talked about axons for a second. Let's talk about dendrites. Dendrites are the receptive region of the neuron. Remember, they can only receive, they cannot send any information. So a dendrite is going to reach out just like the axons are, but typically they're going to be smaller and more numerous. Also, you'll notice that they can convey graded potentials or incoming messages towards the cell body. This is a beautiful example of what a neuron cell body looks like. You can see the dendrites spiking off of there. There is also tons of them. If you were going to judge this as being a typical neuron where the largest or the biggest piece is always the axon, this would most likely be our axon coming off right here. All right, the axon structure, of course, does have a hillock, and if I click back, we can see this wedge-shaped piece right here. This would be the axon hillock. The axons typically are going to branch, and what I mean by branching is they will actually split into individual pieces, and this is what gives us those axon terminals. Remember that the term terminal means the end, so this is the end of the axon, and when we branch, it helps them to reach out or make contact with multiple different cells. In this case, we're going to be looking at multiple different cells. One we talked about before, look at, for example, we talked about neuromuscular junctions. So a neuromuscular junction, this connection point would be made by an axon terminal and the junctional folds of the sarcolemma. This is what this axon terminal would do. You can see they can branch up to 10,000 different branches depending on how many nerves are there or how many need to infiltrate the muscle tissue. So what we find is that these axons are the large structures. They are those elongated, bundled up information carriers that go to all the different parts of your body. The functionality of the axons, they are the conducting region, but as I said before, they can send and receive information. That's right, send and receive, whereas the dendrites were only receptive. This axon will transmit information along what's called an axolemma. Now, we've talked about plasma membranes, or the outside of these cells in our bodies. And when we got into the muscle cell, we talked about sarcolemmas, being the term sarco meaning muscle, and so that outside area was called the sarcolemma. Now, the outside of the axon portion, this is called the axolemma. The axolemma is the plasma membrane that covers the axon, the process that leads down. Now, in every single one of your axon terminals, what you're going to find is a bunch of neurotransmitters. And if we remember what neurotransmitters were, they are the language of the nervous system. This is the language of the nervous system. So this is how they communicate with other cells. And each one of them will have the ability to transmit information, send information, in order to allow for communication with any cell type that it's connected to. Now, the neat thing about these axons is that they don't only have to send information in one way. Because they can send and receive, they can move in both directions. So what you'll notice is something called anterograde or retrograde. If it's anterograde, that means it's going away from the cell body or sending information. If it's retrograde, that means it's toward the cell body, and that means it is bringing information in. That's the axon. All right, so we're all often going to see the term myelin sheath. Now, what a myelin sheath means is simply just insulation. And as I shared before, insulation is the pieces that are wrapping around the outside. And we know that those outside wrapping pieces were called neuroglia. And that means that anything that's neuroglia also kind of classifies itself as a myelin sheath. You'll notice these are protein lipid substances that segment themselves or create this insulated sheath around the outside of the axon or around the outside of the cell itself. And so what they're doing is they're trying to protect and electrically insulate the axon. Pretty simplistic, right? We're protecting and taking care of the axon itself. The non-myelinated fibers, or what is called a non-neuroglia covered cell, is going to show that their impulses are not going to be sent as effectively, and they're not going to be able to be sent as clearly. And one of the main reasons for that is simply because think of what an insulation does. The term insulation is made to help whatever is inside of it stay that way. If we're talking about insulating a house, we want to keep it warm or cold inside. If we're talking about insulating a wire that's in the wall, we want to make sure that that electricity or the transmission doesn't get out. Same sort of case here. So if we have non-myelinated fibers or non-insulated fibers, they're not going to send information as clearly or as effectively as the others would. So for the majority of our axons in the body, they're going to be covered by those Schwann cells, right? Because we said that they were outside of the PNS. They were wrapping themselves around the outside of these cells and helping to protect the cells or helping to send information. So this myelin sheath, I like the way your book puts it. It wraps around in a jelly roll fashion. Okay, this jelly roll fashion just looks like this. It starts wrapping itself, and what it does is it creates these layers. And if you'll notice, once we get down here, we have multiple layers, with the outside being this thicker portion there. This layering helps protect the inside structure, which is, of course, the axon, where it's transmitting or sending information and sending and receiving. This is a beautiful picture of what an axon would look like with a cross-section view. You can see it's magnified at 24,000 times, which is an immense calculation. You know, we're really pushing the limits of the microscopy here. This is multiple layers. If you count those rings around the outside, there could be 15 or 20 rings there, and then you'll notice that the outer collar, the cytoplasm, is kind of pushed to the outside. This is a great insulation for these axons. All right, I've mentioned the myelination within the PNS, but there's a couple other things that I want to show you. First of all, there were some gaps that are found in between those little bubble-like structures of the Schwann cells, and those little gaps are called nodes of Ranvier. They're just tiny little gaps where the axon collaterals can emerge. The other thing that we're going to notice is there's something called saltatory conduction that's going to come up, and it allows for that to kind of spark up the signal as it's being sent down there. If they're non-myelinated fibers, of course, they don't have the myelation that's really good there, and typically they're not going to have the same type of gaps and what's not. As far as myelin in the CNS, we've been kind of talking about the PNS very much. So in the CNS, excuse me, what we're going to find mostly is that the axons are going to be wrapped up together and kind of bundled. And so what they end up looking like when we have a bunch of myelinated axons bundled up together, that if you remember what they're called, in the CNS it's called a tract, but when there's a bunch of myelinated axons bundled up together, that is what makes up the white matter of your brain. Now hopefully we get a chance to see this in the lab when we do our brain dissection, the difference between white matter and gray matter. The white matter is myelinated axons. The gray matter portion, mostly neuron cell bodies. Remember, 99% of our cell bodies are found in that CNS structure. All right, 3 different types of neurons. We've got multipolar neurons, bipolar neurons, and unipolar neurons. If they're multipolar, they're the ones that I showed you earlier. These are probably the most important. They're the ones you're going to see mostly on your exam. A bipolar is only 2 processes, and a unipolar is only 1 process. Now, what I mentioned earlier is that anytime you have an axon, the largest structure that comes off is always the axon. So therefore, if there's only 1 structure that comes off, that is called an axon. If there are 2 pieces that come off, the biggest one will be an axon, the other one will be a dendrite. And then multipolar, always 1 axon, and then a whole bunch of dendrites. Classifications of neurons, grouping in the direction of the nerve impulses traveling relative to the CNS. And you're probably thinking, okay, well, what does he mean exactly by that? I'm just talking about which way does it send information. Now, I already gave you the information that goes along with this, so this should be a quick review. Sensory going towards the brain, motor going away from the brain, and then interneurons are going to be those integral part where integration takes place. That is going to be kind of the control center inside. Here's what they look like: sensory, motor, and interneurons. Sensory transmitting impulses away from this, or towards the CNS, toward the CNS. Motor is carrying impulses away from the CNS, and then interneurons are the ones between. And as I mentioned before, 99% of the body's neurons are found in the CNS. Pretty simple. So let's talk about how we send signals then. We already kind of been over this because we've talked about action potentials and you've seen how information is sent, especially at the neuromuscular junction. So I want to just give a quick explanation of it. Neurons are very highly irritable, and so they respond to stimulus really well. And anytime we are sending or receiving information, it is going to be generating what was called the action potential. Now, if you remember, an action potential was going to be generated in a couple different ways. Most of the time, it's going to be stimulated by electrical, or stimulated by some type of chemical, or stimulated mechanically. So what we use are those channels. Now, leakage channels and gated channels we saw way back in Chapter 3. No surprises here. Leakage channels are always open. Gated channels need to have something to change the shape. What causes that to happen? Well, of course, chemicals, voltage, or mechanical, right? We just said. Chemically gated channels are going to be those ligand gated channels. They are utilizing some type of binding of a chemical. In our case, mostly neurotransmitters, right? Neurotransmitters. Then there's the voltage gated channels, which use electricity, and mechanical, of course, it has to have a physical deformation in order to change its shape. I'm going to skip over the pictures and just talk a little bit more about these. Because the gated channels, we know that when they're open, they are allowing for signals to be sent through. They allow for ion passage to go through, and we saw these mostly as those sodium-potassium channels, and so that's where we're going to spend some more time. All right, in the drawing of our action potential, if you remember our chart that we drew up on the board, there was going to be the straight line across. That is our resting membrane potential. The resting cell, the potential across the resting membrane portion. Typically, it's going to be at about negative 70 millivolts. Now, as we increase that, it can actually change in voltage from 40 to 90. But remember, when we make a change in our resting membrane potential, it's usually going to polarize itself about 100 millivolts. That means if we're starting at negative 70, it's going to change up to about positive 30. And if you remember our action, we drew up and then down and came back across. So it's going to go up to about positive 30, then come back down. That action potential is always generated by the difference between the ICF and the ECF. If you remember, we're always going to be higher in sodium in the outside, higher in potassium inside. That causes our change to take place. ECF has a higher concentration of sodium, ICF a higher concentration of potassium, and so that is what allows our cell to make those changes. Now, when we talked about leakage channels, if you remember, leakage channels mostly are going to be susceptible to potassium. So if we're typically thinking that the leakage channels are more susceptible to potassium, that says that potassium is going to control resting membrane potential. It allows for the resting membrane to kind of maintain its certain levels. So more potassium diffuses out than sodium diffuses in, and that causes that cell to create this negativity or a more negative environment inside. Whereas when the sodium pump is working, it's going to pump in more sodium than is pushing out potassium, and that's what causes the change in the membrane potential. So as it changes, it goes through those phases. We talked about depolarization. Please read through the information about depolarization, about hyperpolarization, and about repolarization. And that's what will give you all the information as to the steps that we go through. If you remember back to graded potentials, they were just short-lived. They didn't reach the threshold, and so typically they don't really count for much, and we don't really spend too much time on them. In action potential itself was just the way that we send information. Here is a brief reversal of membrane potential with a change in voltage, typically of 100 millivolts. That is your action potential. Here is the action potential chart, and I'm actually going to skip down a little bit to a different chart because I want you to read through all that, but here's what we're getting to. Here is my action potential chart that you're going to see on your exam. You'll notice there's a resting membrane potential here. Then we go through our depolarization phase, where I have an influx of sodium. Then I've got my repolarization phase, which is going to be my efflux of potassium. Then I go into a hyperpolarization phase because too much potassium got out. Then we come back to a resting membrane potential because of the leakage channels allowing potassium to come back in. This brings us back to a resting state where you'll notice we're back at number 1. This is back at my resting membrane potential.